Thanks very much, Micardle, for organizing again a great conference. My name is Jan Smeitink, and I'm the founding CEO of Khondrion. And while showing this slide, I would like to thank the organizers for giving Khondrion the opportunity to present our work and showing you our plans for the future. In my talk today, I will concentrate on where we are with our clinical development programs with our lead compound, sondichromanol, also known as KH176. First, the usual disclaimer that any forward-looking statement in this presentation represents our view only as of today and should not be relied upon as of representing our view for the future. Khondrion, ladies and gentlemen, is headquartered in Nijmegen, the Netherlands. Nijmegen is located in the eastern part of our country. We recently moved to this building at the Noviotech campus, and the Noviotech campus is one of the Netherlands' major science hubs, which is a real milestone for us, which will bring opportunities in our own labs and for further collaborations. The focus of everything we're doing is on primary mitochondrial diseases, and our lead product is sondichromanol. Sondichromanol is an orally bioavailable drug with a unique profile. It's a small molecule and can be considered as based on the mode of action, which is redox modulating, radical trapping with anti-inflammatory properties as a pipeline in the product. The results of both the phase 1 and the completed phase 2A showed that sondichromanol is safe and well tolerated. The route of production, so from drug substance, drug product to formulation, is ongoing, and market formulation will be ready at the start of phase 3. Of importance is that sondichromanol has excellent pharmacokinetic properties with high oral bioavailability, and importantly, certainly for mitochondrial disease, has the ability to cross the blood-brain barrier. We completed a phase 2A study which showed significant improvements in mood and attention. And this study should be considered as an early proof of concept study. It was a study performed in Nijmegen, a single center study, like all our studies, randomized, double-blind and placebo-controlled with 100 milligrams twice daily orally of sondichromanol. The study involved 18 patients with the T243 mutation responsible for Miller spectrum disorders. The duration of the study was 4 weeks, and the outcomes were measured after 28 days, both efficacy, safety, and pharmacokinetics. And importantly, we saw significant improvements in clinical and functional endpoints, both on attention as well as in mood. And with 3 independently scored outcome measures, we confirmed the effect on brain parameters. Attention is based on patient testimonials, causing a significant impact on the quality of life, both socially as well as working life. No treatment-related adverse effects were observed in this phase 2A study. So based on these promising results, we designed a phase 2B study, which is currently ongoing and will have readout in Q2 2022. This study, called the Canarex study, is a multi-center study in which the participating centers are localized in the Netherlands, Germany, United Kingdom, and Denmark. Again, a randomized, double-blind, placebo-controlled study, including also a dose-finding arm. This study has 27 adults, again with the T243 mutation, and of importance to better manage patient heterogeneity, we included a cognition-related patient inclusion test. Study duration, again, will be 4 weeks, and the outcomes will be assessed after 28 days. The goal of this study is to confirm the good safety profile observed in the phase 2A, confirm the positive effects in various cognitive domains, now using additional broadly validated computerized tests, with a primary endpoint being the visual identification test. In addition, we will measure parameters in other domains, including hearing, migraine, and fatigue. Patients who participated in the phase 2b trial will be offered to roll over in an open label extension study called the Canarex Trial. This is an open label study with a duration of 12 months in which the outcome measures will be assessed at 3 months interval. The goal of this study, which is ongoing, is to establish longer-term safety profile, gather additional longer-term patient data across phase 2b outcome measures, and assess also complementary clinical outcome measures, including several motor function-related parameters. So with both studies, we hope to reconfirm the phase 2a findings in a larger patient pool. We applied more stringent inclusion criteria to better manage patient heterogeneity and added cognition endpoints broadly validated in CNS indications. Included will be outcome measures that were not studied before, and patients will be rolled over for a long-term extension study. And with this strategy, we hope to create a valuable data package to optimize the phase 3 study design and further support discussions with regulators. Next to these studies in adults, we recently started also the first study in children. This is a phase 2 study which follows first a 4-day adaptive PK study to determine the adult equivalent dose. Again, randomized controlled, now with a primary endpoint on motor function. So brain and muscle are the most important affected organs in patients with Micardle disease. In adults, we focus on the brain. In children, we will focus on the motor function. This study, which is ongoing, is a 6-month study, and outcome measures will be assessed 4 times during the trial period and once during the follow-up. The gross motor function measure, which is the primary endpoint, is an observational clinical tool designed to evaluate changes in gross motor function, which includes lying and rolling, crawling and kneeling, sitting, standing and walking, running and jumping. And this study is supported by a grant from the European Union. So in the next 9 months, a wealth of clinical data will become available. The full results of the ongoing phase 2b study in Miller spectrum disorders, 3 and 6 months interim results from a 12-month open label extension study involving patients that completed participation in the phase 2b trial. And with this, we will have further interactions with EMA and FDA, patient organization and key opinion leaders and other stakeholders to design the most optimal phase 3 clinical trials for both children and adults. So to sum up, sonrotoclax is a small molecule, safe and well tolerated, with a triple mode of action and excellent PK properties. We have an extensive research program ongoing with a completed phase 2a, a near completion of the phase 2b study, an ongoing 1-year open label extension studies in patients with the 3243 mutations, and a recently started phase 1/2 study in children, of which the adult equivalent doses in the age ranges of 5 to 7 years in between have been determined, and the first patients are randomized in the phase 2 part of the clinical trial. And with that, I would like to thank you for your attention.
Thank you very much, Jan, for your presentation. And again, all the molecules in the development programs will be discussed at the end of the session. So please use the Q&A function in Zoom to produce your questions that will be transferred to the speakers after the all lectures. So now I have to introduce Anthony Abbruscato, who is the Senior Director of the Development Program of Stealth Pharmaceutical. So Anthony, now the floor is yours. Thank you.
Thank you, Professor Mancuso. Okay, so hello everyone. My name is Anthony Abbruscato. I'm the Senior Director of Clinical Development at Stealth BioTherapeutics. Happy to join you all today to discuss our development efforts with elimipritide for the treatment of primary mitochondrial myopathy. First, I'd like to provide a brief overview of the mechanism of action of elimipritide. Elimipritide is a positively charged tetrapeptide which localizes to the inner mitochondrial membrane. Through stabilizing its target, a phospholipid called cardiolipin, elimipritide is able to restore the morphology of the cristae, which is disrupted across many mitochondrial diseases, thus restoring the proper function of the electron transport chain, as well as other inner mitochondrial membrane dynamics, such as protein importation and other cardiolipin-dependent processes. MMPower3 was our first phase 3 trial in patients with primary mitochondrial myopathy. This was a global study initiated in 2017 and completed in late 2019. MMPower3 was a randomized, double-blind, placebo-controlled trial, which enrolled 218 patients with PMM. Patients were randomized in a one-to-one manner to receive elimipritide 40 milligrams or placebo for 6 months. All patients had access to elimipritide through a long-term open-label extension following the double-blind portion of the trial. The two co-primary endpoints for this study were the distance walked on 6-minute walk test and the total fatigue score on the PMM symptom assessment, which was a PRO developed by Stealth. MMPower3 was designed as a basket study. While all patients had to have a diagnosis of PMM, the study allowed a variety of underlying genetic mutations. About 75% of patients had a pathogenic mutation in mitochondrial DNA, while 25% had mutations in nuclear DNA. Here's a visual breakdown of the specific genotypes enrolled in MMPower3. Mitochondrial DNA mutations are shown in gray, nuclear DNA mutations are shown in green. Milas syndrome or MTTL1 mutations made up about 1/3 of the mitochondrial DNA subgroup, followed by patients with large single-scale deletions. For the nuclear DNA mutation subgroup, the most common mutations were found in the polymerase gamma POLG gene. Here's the top-line readout for the study. Both co-primary endpoints are displayed, 6-minute walk test on the left and total fatigue score on the right. You can see that both endpoints showed improvement from baseline. However, there was no clinically or statistically significant difference between treatment and placebo. The study did not meet either of its primary endpoints. While this was not the outcome we were hoping for from the trial overall, we knew we had a lot to learn from this data set. Our first step in diving in further was to understand any differences between the genetic subgroups included in this study. To the left on this slide, you can see the results from the 6-minute walk test in the mitochondrial DNA subgroup. Notably, there seems to be a large placebo effect, which was particularly seen in the Milas patient population, with those patients walking about 40 meters more at week 24 than at baseline. When we look at the nuclear DNA subgroup on the right, there appears to be more of a separation between treatment and placebo. Interestingly, there was no observable placebo effect in this subgroup. The patients randomized to elimipritide walked almost 30 meters more at week 24 than at baseline. So we wanted to explore this potential signal further. This is a similar graphic to that presented previously, but this highlights only the breakdown of the nuclear DNA subgroup. As mentioned, the most common nuclear DNA mutations were of the POLG gene. Beyond POLG, a vast majority of the mutations included in the subgroup were also related to either mitochondrial DNA replication or maintenance of the mitochondrial DNA nucleotide pool. So while there is still genetic heterogeneity in this subgroup, the function of many of these genes are related. To further explore this potential signal in the nuclear DNA subgroup, a pharmacokinetic exposure response analysis was performed. This analysis looked at response as defined by 6-minute walk test as a function of plasma area under the curve. You can see that there's a statistically significant correlation between exposure and response. This analysis is particularly encouraging as it provides confidence that the subgroup finding is unlikely to be due to chance alone. The other key takeaway is that the therapeutic dose may not have been fully optimized as the exposure response curve is not saturated. In addition to interrogating the genetic data, we also spent time understanding the impact of different phenotypic presentations on the outcome of our study. The graphic shown on this slide highlights the differences in clinical presentation within POLG-related disorders based on age at onset of symptoms. In our trial, despite the common diagnosis of PMM, there was a variety of other comorbid conditions and syndromes present in each patient's medical history. While I don't have the data shared on this slide deck, we analyzed our endpoints by looking at any association with age and by grouping patients based on certain medical history terms. What we found is that patients with a comorbidity of progressive external ophthalmoplegia, or PEO, had a better response than those with other medical history terms when compared to placebo. So putting those two deep dives together, we arrived at a targeted patient population that we believe will benefit most from treatment with elomiprotide. The graph on this slide represents the change in 6-minute walk test in patients with nuclear DNA mutations involving mitochondrial DNA replication or maintenance with the comorbidity of PEO. And you can see in this subgroup of 32 patients, there was a 45-meter difference between treatment and placebo with a p-value of 0.015. So what does that mean for our program and next steps? We're very encouraged by the signal from MMPower3, and our goal is to replicate that signal in a follow-on phase 3 trial. This upcoming trial will follow a similar study designed to MMPower3. However, we have narrowed the patient population to be more homogeneous based on those subgroup findings. The key elements can be shown on the right side of this slide. We are still including patients with a primary clinical manifestation of myopathy, but enriching for those who have replosome-related mutations and a comorbidity of PEO. And based on the exposure response analysis, we're increasing the dose from 40 milligrams to 60 milligrams to optimize response. And the other key change to note is that we are increasing to a 1-year treatment period, as data from other programs suggests there could be increasing benefit over time. We're excited for this study to be initiated by the end of this year. So in summary, to share some lessons learned from our development experience with elomiprotide in PMM, we have learned some mechanistic insights about elomiprotide, specifically that nuclear encoded control of mitochondrial DNA maintenance and replication may have particular relevance in this patient population. We have learned that both genotypic and phenotypic heterogeneity in this patient population can be challenging to address and can potentially mask a treatment effect. We learned that reliable PRO assessments are still needed for this patient population, particularly to address muscle weakness and fatigue. And finally, we continue to appreciate the collaboration with the Mino community, who have helped us conduct high-quality clinical trials and who have helped us interpret our data to enable continuation of our development efforts with elomiprotide. We look forward to continued collaboration for our upcoming phase 3 trial. And thank you.
Thank you, Anthony, for your introduction. Serenella.
Sorry. Oh, sorry. So let's go on with the Magnus Hansson, that is the Chief Medical Office, VP Preclinical and Clinical Development from Abliva. Please, Abliva.
Thank you very much, Professor Servidei. Do you all hear me and see my screen?
Yes, yes, perfectly.
Thank you. Yeah, so also thank you to the Mitochondrial staff for arranging this meeting and for inviting us. So I'm Magnus Hansson, Chief Medical Officer at Abliva. And at Abliva, our mission is to improve the lives of primary mitochondrial disease patients through innovative drug development and research. So at Abliva, we have a portfolio of innovative compounds. To the left, KL1333, which is a first-in-class compound aimed to correct the NAD plus NADH disbalance that follows from dysfunctional mitochondria in mitochondrial disease. Here, the target is multisystemic mitochondrial disease with a specific focus on pervasive fatigue and muscle weakness. Our second compound, MV354, is another type of compound. It's a prodrug of succinate. It's also a first-in-class compound. Here, the target is Lee syndrome. But first, focus on KL1333. A highlight of the journey of this compound so far. And this journey has really been informed by patients in order to reach patients. This is a highlight from a published study in MELAS patient fibroblast, exploring the mechanism of action, showing how improved NAD plus over NADH ratio improves cell energy status, mitochondrial function, through upregulation of what is called mitochondrial biogenesis pathways to build up new components of mitochondria. So how do we translate improved energy production in the cell to an endpoint which is valuable for patients? So in the program, as we've heard over and over again, that the lack of energy that patients feel, this pervasive fatigue, is really important. And as mentioned in the previous talk, we really need validated tools to assess this. So we set out to validate a patient-reported outcome, looking at fatigue, lack of energy, and the impact it has on daily activities of living. So we performed an interview study, 2 sets of interviews, to validate a set of questions which were already validated in other disease areas. And the result is that we now have a comprehensive and regulatory viable mitofatigue short form. And presentation and discussion of these results is available on YouTube. It was released during the Mitochondrial Disease Awareness Week. You can find it on YouTube if you search for fatigue in primary mitochondrial disease. The first actual efficacy data from patients was reported earlier this year for KL1333. This is data from a small phase 1b trial. And here we saw signals of efficacy that are seem to be clinically meaningful when we look at the magnitude of effect and comparing those patients who received active compound KL1333 versus placebo. And the 2 type of outcome measures we had in this trial were a patient-reported outcome of fatigue, as well as assessment of muscle function and muscle weakness in the 30-second sit-to-stand test. So this has been very encouraging and informative of our next step, which is a phase 2-3 trial, which we have designed. So if it's successful, it has the potential to be the only remaining trial before market approval. So a short highlight of that trial, we will focus on patient-reported fatigue using our validated short form for fatigue, as well as the same test from the previous trial, the 30-second sit-to-stand test. This trial will include adult patients with mitochondrial DNA-related multisystemic disease, and the trial will last for 12 months. Our second compound, MV354, is now completing all the preclinical data that is required to take the compound into clinical development. And we expect to start phase 1 trials early next year. And this follows from a successful first regulatory interaction with this compound with the UK authority, MHRA, just a couple of months back. So MV354, this is a prodrug of succinate. So succinate is an energy metabolite that enters the electron transport chain at complex 2. Thereby, it has the potential to bypass the deficiencies caused by complex 1 dysfunction, which is common, for instance, in Lee syndrome. We have evaluated this compound in many different disease models. So this is recent data from the genetic Lee disease model in animals, the NDUFS4 model, where we see a decrease in neuroinflammation, delay in the presence of clinical and motor symptoms, and also improved weight gain development. So we look forward to continue the development of our programs with collaboration with the mitochondrial disease community.
Thank you, Magnus. Thanks for your presentation. We will discuss at the end. So now it's time to have with us the Madhu Davies, the medical director of Reneo Pharma. Thank you.
Good morning, everybody. It's an absolute privilege to be able to speak to you all today, and I would like to thank the organisers for inviting me. As the professor said, my name's Madhu Davies, and I'm the medical director at Reneo Pharma. I hope that you're seeing my slides properly. I'm already running into some technical difficulty with advancing them, so just bear with me.
It works perfectly.
Thank you. So before I start, I need to provide you with the usual disclosures, which are at the bottom of the slide. In particular, our drug, REN001, is an investigational drug and is only available within our research programme. The mission of the company is to try to do what we can to help patients with metabolic myopathies, particularly with regard to their endurance, fatigue, and overall well-being. The organisers very kindly invited me to talk about our research programme in mitochondrial diseases and gave me 8 minutes to do this, so apologies if this is a little bit brisk. First of all, our molecule, REN001, what is it? Well, it's a bit of a mouthful. It's a peroxisome proliferation activated agonist, or PPAR delta. And so what? What does that mean? Well, it's a member of the family that regulates cellular energy generation. As you might expect, it's therefore present in tissues that require a lot of energy, such as muscle, kidney, brain, and liver. And the receptor switches on when the body needs more energy and calls for that energy to be delivered. And it works in 3 main ways, as far as we know at the moment, which is really interesting. First of all, it increases the transcription of genes, which are central to mitochondrial function. So it's trying to make the mitochondria work better. It appears to drive the production of new mitochondria, which could be quite interesting. So mitochondrial biogenesis. And critically, probably most importantly for this audience, it increases fatty acid oxidation and therefore the production of energy by the cells. So on that basis, we're investigating this drug in a number of mitochondrial diseases, in particular, primary mitochondrial myopathies, about which we've already heard a lot this morning. But we're also looking at long-chain fatty acid oxidation disorders and Mcardle disease. And as you can see from the graph, our primary mitochondrial myopathy program is currently the most advanced, and we're in a phase 2 study that I'll talk about in a moment. but we're also in early patient studies with both long-chain fatty acid oxidation disorder and Mcardle's. So for those of you who have primary mitochondrial myopathy, this slide is very old news. But for those who are not so familiar with the condition, primary mitochondrial myopathies are very rare disorders and caused by the mutations that colleagues have been describing already this morning, either to the mitochondrial DNA or to the nuclear DNA. And bottom line, these mutations stop the mitochondria from generating energy in the way that they should. And again, as you would expect, you're going to notice that most in the tissues that require lots of energy. So again, muscle in particular. Patients with primary mitochondrial myopathy will recognize what I'm going to say very clearly. Debilitating fatigue, myopathy, exercise intolerance, muscle pain, and lack of endurance. And some patients tell us that they can't even play with grandchildren or go and do their shopping without being severely impaired. And that's horrible, isn't it? Because we all take these things for granted otherwise. Critically, there are currently no approved therapies. So I'm delighted to hear that so many of us are working in this area, hopefully to bring an improvement to these patients. And patients currently rely on over-the-counter vitamins and supplement cocktails to try to improve mitochondrial function. So as I say, I'm really encouraged by all the research that's going on in this field now. So what are we doing in primary mitochondrial myopathy? Well, we completed a phase 1 patient study which demonstrated that the drug was safe and well tolerated as far as you can ever tell at that stage. And that allowed us to proceed to a phase 2 study, which is ongoing now. That study is essentially checking the activity of the drug against a placebo. And what we're interested in is the change from the beginning to the end of the study in the distance that patients can walk in a specific time. Critically, we're also very interested in how the patients feel and how they function, because it's all very well helping a disease to be better, as it were, if the patients don't feel any better. So we're very interested in that aspect of this drug as well, whether patients feel better. Who can participate? Well, adult male and female subjects, and by adults I mean 18 years old, and they need to have primary mitochondrial myopathy as defined by the International Workshop, which was published by Professor Mancuso here on this webinar. Patients need to have defined mitochondrial DNA mutations only. We had to start somewhere, so that's where we decided to start. And they need in particular to have primary mitochondrial myopathy, which is characterized by poor exercise ability or active muscle pain. Because we're assessing how patients walk and how far they can walk over a set time period, naturally the patients need to be able to walk, and they need to be able to walk on their own without another person helping them. Although they can use a walking stick or other walking aid if that's necessary for them. Importantly, women may participate in the study if they wish, as long as they're prepared to use highly effective contraception. This overview really just covers much of what I've already said, but in one handy place. So it is a multi-center, multinational, double-blind, placebo-controlled randomized study in which patients will either receive dummy drug or active drug, and none of us will know because it's a blinded study, and they'll take the drug for about 6 months. Importantly, from the point of view of the audience for this webinar today, there are 5 sites in Italy. I'm delighted to be able to say that. I've already explained who the likely patients are in terms of the profile, and I've already explained the outcome measures, but they're just there for you for easy reference. A question that we're asked routinely, because sites around the world are widely spread, is, are travel expenses covered? And the simple answer to that is yes, they are. I'm absolutely thrilled to be able to say that further information about the study can be found on clinicaltrials.gov, but also on the Mitochondrial Italy website. And thank you very much to Mitochondrial Italy for helping us with that. Oops, apologies. We are active in other areas as well. And in particular, we are running studies, as I mentioned, in long-chain fatty acid oxidation disorder. And we have a phase 1 patient study currently running in that area. We're interested in the safety and tolerability of the drug at 12 weeks in patients with long-chain fatty acid oxidation disorder, but we're also interested in exploring changes in certain clinical outcomes, such as 12-minute walk test, or changes in symptoms, which tell us how patients feel and function, which, as you've gathered, is very important to us at Reneo. We're also working in Mcardle disease, and here's a slide just summarising a little bit about that, because it's such a rare disease and so unusual. So in this condition, patients cannot break down the glycogen that's installed in muscles, so they lack the energy to get going and have this absolutely debilitating lapse in energy production after a very short time. And I'm talking seconds to minutes. And this gives patients horrible, horrible acute muscle pain, very severe fatigue, and an elevated heart rate. Critically, from the point of view of their health, not only are they unable to get going, climbing stairs safely, or getting out of the car to go into a supermarket without some discomfort, but it can also result in severe breakdown of muscle or rhabdomyolysis, which leads to hospitalization because of acute kidney failure, and of course in really severe circumstances could be fatal. And horribly, there are no treatments for this. So we're very interested in pursuing this as well to see if our drug makes any difference. And what we're doing there again is a phase 1b clinical trial, very similar to the one I've just shown you for fatty acid oxidation disorder, where again, we'd like to assess the safety and tolerability of treatment with our compound over 12 weeks, as well as how patients feel and function. So with that, I would really like to say how grateful we are to the patients and wider Mitochondrial community, because essentially we wouldn't be able to do any research without you all. And with that, I'd like to say a big thank you. Thank you very much.
Thank you very much, Madhu. And we can go on with Salvatore Funiciello, Medical Director Italy, the Zogenix. Please, Salvatore.
Can you see my screen?
Yes.
Thank you, Professor. Thank you for your presentation. Before I start, I want to thank the organizer for giving us the opportunity to share our experience in clinical trial for rare disease such as mitochondrial disease. As you mentioned, my name is Salvatore Funiciello. I'm the medical director of Zogenix, a new company that is a small company. We are, since our beginning, fully involved in the development of a drug for rare diseases. Our organization, of course, is first mainly located in US, in the San Francisco Bay Area, but we have also additional sites in Europe, like Italy, Germany, France, UK, and we have also a partnership in Japan. At the moment, we are all over the world, only 180 full employees. We mainly started to work on rare severe epilepsy, but we, since a couple of years ago, started to work on mitochondrial disease, and in particular for thymidine kinase 2 deficiency with the program MT1621. Few words on the Tk2 deficiency. You know better than me that is an ultra-rare genetic disease. The mutation of the Tk2 gene in the genome in the nuclei can impair the functionality of the mitochondria in the cells. So this can present a lot of variety in terms of showing the disease. So the patient can start to present severe proximal muscle weakness, and this is a progressive disease and can lead to death approximately 50% all overall Tk2 deficiency population. The muscle weakness, you know better than me, is progressive, can have slowly or a rapid progressive symptoms. And the age of onset could be at any age. So we have early onset and also adult or late onset. And like most mitochondrial disease, the Tk2 deficiency is very difficult to diagnose. We are developing this molecule that is the MT6021, that is a nucleoside therapy in equal part of DCT and DT, oral solution in powder. And this drug has shown to make improvement in the salvage pathway of the residual activity of Tk2 and restore the mitochondrial function. The study that we are having in our company, we have three main studies. One is close to start, and one is already completed. The first one is the MT6021-101. It was a retrospective study completed. We managed 38 patients, adult and pediatric. All of them were genetically confirmed with Tk2 deficiency and were treated with chemical grade nucleosides. In this study, we showed that there was an improvement of survival rate when compared to untreated patients. There were also improvement in motor, respiratory, and feeding function. There was also a stability or improvement in patients that had a rapid or a slower progressing disease, and the treatment was generally safe and well tolerated. Now we have another one that is the MT6021-102 study that is a phase 2 prospective, in this case, study open label, and is a continuation of the first study I spoke before. 47 pediatric and adult patients, 35 are coming from the 101, and we have the same approach of the first study, but this is more concentrated on the dosing approach of the drug. The baseline, of course, will be linked to the retrospective study that we have completed so far last year. There is also another study that we want to carry on soon, that is the MT6021-104, is a phase 3B single-arm study in Tk2 with pediatric and adolescent patients who are naive to the treatment with MT6021. We are going to enroll 16 patients. The age of the onset will be less than 12 years and less than 18 years at time of consent. All of them genetically confirmed with Tk2 deficiency. There will be a 3 months run-in without any treatment, followed by 12 months period with treatment, and plus an extension phase of 12 months more. The sites will be based in UK, France, and US. The endpoint of this study is the proportion of subjects that are acquiring motor milestones not present at baseline after 12 months of treatment. This is probably the last slide I will show you. We have tried to give a picture of what are the pitfalls and the challenges that the company so often working on this kind of diseases facing every day. You know, for example, speaking about Tk2, that the genetic testing is play a crucial role in this disease, but so often when the genetic tests are ordered, is used incorrectly with the wrong test, the wrong DNA panel that not include Tk2. And there is also, in terms of diagnosis, that normally healthcare professional, not the specialist as you are, so often they don't suspect mitochondrial disease and especially Tk2. And there is also a limitation that to carry on clinical trial on Tk2, the genetic test is required to make the patient enrollable in the trial. In terms of the disease, you know, the disease is very heterogeneous. the variety of the symptoms is very high. So this is also another difficulty that we can face in structuring a clinical trial. And we can say also that the benefit for one patient will not be relevant for another patient. And also the difficulty that we have very small population to work and to enroll in our clinical trial. In the clinical meaningful endpoints, I think the most important is the gap in understanding the disease, and also that the natural history of the disease is not well documented. And also in terms of regulatory with the agency, the regulatory pathway is typically unknown. There is a lot of work still to do. I think this is the last one, and I want to thank you all for the attention.
Thank you, Dr. Funiciello, for your presentation. Now it's time to introduce John Campbell from the BioTherapeutics. He's the head of the clinical development program and already awake for us in the US. Thank you, John.
Thank you very much, Professor Mancuso. Thank you to Mitochondrial for inviting us to speak today at this important conference. As Professor Mancuso mentioned, let me just so as Professor Mancuso mentioned, I'm from BioTherapeutics. My name is John Campbell, and I'm head of US clinical development. Manovia is a clinical stage biotech company with headquarters in Haifa, Israel, and offices in Boston. We're developing a therapy called mitochondrial augmentation therapy for the treatment of primary mitochondrial diseases. We feel at Manovia that harnessing the power of mitochondria itself to treat mitochondrial diseases is a very sound approach. Just to briefly run through in the time we have of this technology, mitochondrial augmentation therapy is a novel cell technology platform where autologous hematopoietic stem and progenitor cells, HSPCs, are augmented ex vivo with mitochondria obtained from donor cells or tissue. It's a multi-step process that begins with producing qualified mitochondria from a donor. In this case, for non-inherited mitochondrial diseases, the donor is the mother, and so that is syngeneic donor, blood-derived. And step two is a collection of hematopoietic stem cells from the patient. After mobilization, these cells are collected via apheresis, and CD34 cells are isolated. And then in step three, we augment those CD34 cells collected with a qualified mitochondria from the donor in a proprietary process to create the investigational product, the NMV-BM-BLD blood product. And then we reinfuse that IP back into the patient, and no conditioning is required for the process. I'd like to draw your attention to a really exciting publication from our team, our R&D team at Manovia. It's published in Nature Regenerative Medicine very recently, and the lead author and the principal investigator of our trial, Elad Jacobi, and team at the Sheba Medical Center. It explains the science behind mitochondrial augmentation therapy and the process of augmenting HSPCs with normal exogenous mitochondria. In that paper, brief summary is a dose-dependent effect that we've seen. We see superior human engraftment in non-conditioned mouse model. We were able to demonstrate durable engraftment in a mouse model. And we feel that these preclinical results are very encouraging and support further development of MAT as a disease-modifying therapy for primary mitochondrial disorders. I'll talk briefly about two programs that we have. First, the compassionate use or expanded access in the United States. In this program, there are 7 patients in total treated under compassionate use at the Sheba Medical Center in Israel. The population included 4 patients, pediatric patients with Pearson syndrome, 2 current Sayre patients, and 1 patient with Leigh syndrome. You can see the ages range from a minimum of 2 to 17 years. And the patients came from multiple countries, including Israel, United States, and Russia. Patients were treated in the interval of time from 2017 to 2020. Patients were followed for safety and effectiveness per individual protocols approved by the appropriate authorities with a long-term safety follow-up up to 4 years. Consistent with our talk today, we did experience impact of this program from the COVID restrictions, which translated into delayed visits, extended visit windows, and we employed remote assessments where necessary. The final in-person patient visit is expected this month. We are looking at the data as this open label primary safety endpoints, and the manuscripts are being prepared. There are some positive preliminary signs of body system improvements, Pearson being a multisystemic disease, primarily manifesting in the bone marrow with bone marrow failure, and leads to also some neurologic involvement down the road. And these patients also frequently have exocrine pancreatic dysfunction. So far, the safety and tolerability profile has been favorable, which is very encouraging. We also have a clinical trial, 001. In this trial, 5 participants were treated. That was amended from a number of 7, which was planned, and also to accommodate remote assessments secondary to the COVID travel restrictions. This trial is an open label, single dose, proof of concept study conducted under an IND. The primary endpoints are the International Primary Mitochondrial Disease Scale, IPMDS, developed at the Raban under the direction of Jan Smeitink on this call, and also safety being a co-primary endpoint as well. The secondary endpoints, there are many, but they include, in summary, laboratory measures across the different body systems, as well as, very importantly, muscle function, growth, strength, and other established measures to correlate with the primary endpoint, including the PETI. The primary period of evaluation is 1 year post-treatment with long-term safety follow-up. The patient population in this study is 4 Pearson syndrome patients. The design was all Pearson patients. However, subsequently, it was learned that 1 of the patients actually had current Sayre syndrome. The average age in this study is 8.5 years, with a minimum of 4.7 to a maximum of 12 years. Participants came from the United States, Norway, and Italy. Their treatment dates range from third quarter 19 to second quarter 2020. As we experienced in the compassionate use, and as many folks have experienced, the operational impact of COVID was significant, but we mitigate these where possible with remote and virtual assessments. Database lock has been achieved, and the statistical analysis is underway. We plan a manuscript and this study will further inform our future clinical development efforts for sure. So challenges and how we address those challenges. Pearson syndrome is a very rare disorder, and typically the patients need to travel to the investigational centers, in this case, Sheba Medical Center in Israel. Of course, COVID imposed several interruptions there at the government level, at the hospital level, but also importantly at the patient level, considering the range of patients and the severity of disease. These patients had safety considerations and individual preferences with respect to their willingness to travel for assessment. Also, the clinical trial endpoints that we chose, particularly IPMDS, some can be administered remotely, but it also requires professional assessment, which we needed to work around. COVID extended our visit window intervals, created some missing data, and also, we wanted to make sure that the patients, the safety and welfare of the patients in the trial was our primary consideration. In mitochondrial disease in general, certainly in Pearson syndrome, objective clinical trial endpoints capable of being applied in the heterogeneous populations that we study and are trying to help has been a challenge. We've heard that from prior speakers, and continues to be a challenge with respect to finding the right population for your study that you can discern a treatment effect. Also, in these rare disorders, in many cases in mitochondrial disease in general, there's a paucity of natural history data to help guide you with respect to the design of your study and to understand and characterize treatment effects that you see. To address these challenges, I think some of the lessons we learned really is to maximize the ability to conduct remote and virtual assessments. You need to build flexibility into the protocol with respect to the visit schedule and protocol assessments. Try to minimize the complexity of the trial, minimize the patient burden to the extent that is possible. And again, these remote visits can include home visits virtually with the principal investigator, as well as in-person visits at the nursing level for vital signs and various assessments, but also potentially at the local HCP level, and certainly at remote laboratory for laboratory values and assessments as well. We have looked at the IPMDS in this study as an attempt to use a multi-domain endpoint that is capable of going across this heterogeneity we see in Pearson syndrome and other primary mitochondrial diseases, and we continue to explore that and explore means of gathering or generating natural history data that is suitable to help us characterize the treatment effect. And we're doing that with partnerships with patient advocacy groups, academic partnerships, and the use of novel technologies such as AI to access laboratory values from the patient records. So in summary, MAT, mitochondrial augmentation therapy, is a novel cell therapy under investigation as a treatment for primary mitochondrial diseases. Our data from the compassionate use in the clinical trial are being analyzed currently, and the manuscripts are in progress. These data will inform us with respect to future clinical development. We are very encouraged by the favorable long-term safety profile of MAT, There were no SAEs in any of the patients that were attributed to the investigational product by the investigators, and the adverse events were predominantly due to the apheresis and mobilization procedure that is required for MAT that occurs prior to administration. We also continue to try to understand the science behind mitochondrial augmentation therapy and our ability to harness the power of the mitochondria as a treatment. And this is the foundation for our future development as well as our clinical data. The operational impacts of COVID on our efforts was significant, but we minimized those impact to the extent we could and maintain patient safety and the needs of patients in our trials. We learned some lessons with respect to protocol design, as I mentioned, and of course, the community as well, as Monovi is part of, we have an ongoing opportunity to make sure that we collaborate across the stakeholders in the process and to keep the patient the main focus, which is a key to success. I would like to thank Mitochondrial for the invitation to speak, as well as all the patients who are patiently waiting for solutions to help alleviate their disease. Thank you.
Thank you, Dr. Campbell. And so we can go on with Xavier Lloria, medical lead, rare diseases, Chiesi pharmaceutic. Please, Xavier.
Thank you very much, Dr. Servidei. Thank you to Mitochondrial for inviting me. In the next minutes, I'm just going to present you a little bit our path, our history in developing idebenone for LHON as a rare clinical, disease of the optic pathway. These are my disclaimers. I won't present the specific results, but I'll tell you, what we've been through in developing this drug. And we know, LHON is it's a disease affecting the optic nerve. It's a disease caused by a mitochondriopathy. And obviously the solution is going to be somehow addressing that. Eventually it will end up in a visual deficiency, visual defect, as you can see here. That is going to be severe, and rarely, it's gonna improve in very rare occasions. As I said, there's a problem at the DNA of the mitochondria. So obviously one approach would be gene therapy, would be replace that missing or wrong gene, and then trying to fix the problem there. In the case of idebenone, the solution is actually to bypass the component that is not properly functioning in order to continue with the energy generation pathway at the mitochondria. The drug is at has gone through regulatory approval in Europe, and a group of experts back in 2017 already produced some guidelines on the use of the drug for the management. But at that time, even at the time of the approval, obviously there are a number of gaps, which is absolutely natural when it comes to rare diseases. You cannot pretend to have all the evidence by the time that you get or submit for regulatory approval. And as my previous speaker already advanced, we have a number of challenges in rare diseases. One is the number of patients. It's very limited, let's say, population. So you cannot pretend to have patients for every single study you want to do. And even for a single study, the numbers usually are very small. Another thing is that the natural history of the disease, the knowledge of the disease, especially at the beginning of the development, is very limited. So it's very difficult to plan or to design proper clinical trials. And then this is the case. I think that of all the panel, we are at the, let's say, at the commercial stage. We've gone already through the regulatory approval, And that brings in a new problem, which is the minute that the drug is in the market, placebo might not be used for ethical and for operational reasons. So these are going to be challenges that we had to address. Now, regarding idebenone, that was, as I said, approved in 2015, it's very normal for regulatory authorities to, let's say, impose or demand some post-authorization development, let's say, in order to clarify aspects that are not really clear, and to complete, obviously, the knowledge of the natural history, and obviously, and most important, to confirm that the efficacy is correct. And you will see here that there are natural history studies and 2 studies at the bottom, LEROS and PAROS. PAROS is going to be purely safety. It's a let's say a proactive pharmacovigilance study which has been finished, included about nearly 230 patients, and we expect results by the end of this year. This is purely on the safety side. Now, on the efficacy side, one of the learnings we've had from all the development has been what to measure, how to measure, and when to measure. And here we can see at the bottom what the international experts showed at the consensus meeting, in which it is not the same when the patient is in the subacute dynamic than when the patient is at the chronic stage. So we should take that into consideration when analyzing and when including patients. Also, the therapeutic objectives. It's not simple to measure just magnitude of changes. It's better, in our opinion, and we've learned that, to go for let's say outcome measures. Establishing a threshold and then seeing if the patient goes over that threshold or not. And then obviously 2 important things. It's not all about recovery. In this disease that you get a very severe visual decline, it's not all about returning vision to a perfectly normal stage, but also avoid the deterioration. And here is what we show, and this is what when you read eventually the results of the study, you will come across these endpoints which are important. And as I said, we will be looking at the patients less than 12 months since onset and patients more than 12 months from since onset up to 5 years. Now, another important thing about this study, LEROS, is that it fills in the gap, and this is one of the pitfalls of the development that we had with idebenone, is we had the perfect study at the beginning, RODOS. Here we have fully double-blind, placebo-controlled, etc. But at that time, the knowledge of the disease, was not very clear what to measure and how to measure and when to measure it. And another thing that we learned a posteriori is that 6 months of therapy was not enough to see everything. So then, a little bit later, we had this real-world experience, which is the expanded access program, that provided, yes, longer-term duration. So we know more, but no control. So LEROS in some way tries to cover that gap. And when you see the results eventually, you will see that here we are having an interesting thing is we cannot use placebo. So we have to do something in order to get a comparator. And this is something agreed with the authorities, which is have a look at the natural history of a number of patients with the same criteria, with the same efficacy endpoints, with the same variables as you would be using in the LEROS. And this is what you're going to see when you see the results. You're going to see a natural history control group that has been matched with the population of LEROS. This is something that in due time it will be explained. But this is going to bring, and the results which have already been released in a press release, the positive primary endpoint, will show how this method really brings a good approach to find further data when it comes to post-authorization studies. LEROS, just on a nutshell, is a 2-year study with the drug at the approved dose, and it has included 199 patients in countries in Europe and United States. So, and just coming to an end of all these challenges and pitfalls is what we know now, and what LEROS is going to bring is a number of new pieces to this puzzle that it is not clear, that should help new development of new drugs. So we know now that we have to profile better. The length of treatment should be longer than initially expected. The responders can be from less than 5 years. I mean, patients up to 5 years could still be enjoying a potential beneficial effect. But still, we're still left with questions, and questions that obviously this study will not be able to address, which is, what happens in patients beyond 5 years? Can we prevent development of the disease in those patients with the mutation? And now with the new developments and knowledge about the nuclear mutations, I mean, what will be the best approach? And well, I thank you everyone for your attention, and I'll be here for questions later on. Thank you.
Thank you, Xavier, for your lecture. Now we have a video, because unfortunately the Mcardle Disease from GeneSight cannot attend the meeting, so I introduce this video from GeneSight Biology.
I would like to warmly thank the organizers for the invitation to present a few insights of our work. Firstly, one word on GeneSight Biologics. GeneSight Biologics is a biotech company developing gene therapy in inherited retinal diseases leading to blindness. We have two main projects using two technology platforms. Our lead program is the development of a gene therapy in LHON. We have another program in retinitis pigmentosa, and we are at early stage clinical development with a first-in-human study which is ongoing. GeneSight was founded in 2012, and the headquarters is based in France. I would like to focus this presentation on Lumivoq gene therapy development in LHON. We developed the technology of gene therapy to overcome genetic mutations in the mitochondrial DNA. The gene therapy Lumivoq specifically targets the 11778 mutation on the ND4 mitochondrial gene. Lumivoq is a recombinant adenovirus type 2 containing the wild type, the healthy mitochondrial ND4 DNA. Lumivoq is injected via intravitreal injection, as it is the best way of administration to reach out the targeted cells, the retinal ganglion cells. Once injected in the eye, the ND4 DNA is transcribed into mRNA in the nucleus. Then the two mitochondrial targeting sequences actively shuttle the ND4 mRNA to the mitochondrial surface, where a functional ND4 protein is synthesized and translocated inside the mitochondria to restore energy production. What are the key milestones for Lumivoq? We filed for EMA registration in September 2020, based on the results of two pivotal studies. We will then be moving to BLA for US registration in Q2 2022, based on the results of a third pivotal study designed with the FDA. Let's move on to Reflect, which is our third pivotal study. Reflex Study has been designed in close collaboration with the FDA and is under special protocol assessment. The FDA requested to test the bilateral treatment of Lumivoq, as LHON is a bilateral disease, and to compare the visual outcome between eyes of different subjects. That is why the pivotal study designed with the FDA has different design from reverse and rescue studies. Reflect is a two-arm study. In one arm, patients were bilaterally treated with Lumivoq, and in the other arm, patients got one eye treated with Lumivoq and the other eye treated with a real intravitreal injection of placebo. The primary endpoint is the difference in change of visual acuity between the second eyes treated with Lumivoq and the second eyes treated with placebo. Let's move on to the results with evidence of a dose effect. Here is the representation of a change from baseline of BCVA over time by eye groups. In red and orange, the treated eyes from bilaterally treated patients. In red, the first affected eyes, and in orange, the second affected eyes. In green, the eyes from unilaterally treated patients. In dark green, the eyes treated with Lumivoq, and in light green, the eyes treated with placebo. The three groups of eyes treated with Lumivoq have a statistically significant improvement versus baseline. The placebo eyes in light green show also an improvement, confirming the contralateral effect of Lumivoq, but this improvement is more modest than the improvement in directly treated eyes. We are now looking at the change of BCVA from nadir at 1.5 years. Bilaterally treated patients got an improvement of 19 and 16 letters in their first and second affected eyes. Unilaterally treated patients got an improvement of 19 letters in their first treated eyes and 13 letters in their placebo eyes. The improvement is compelling for treated eyes and again less pronounced for placebo eyes. Other ways to look at efficacy are equally important. We looked at the patient responder rate at 1.5 years. Firstly, we looked at the switch from off-chart to on-chart, which is clinically meaningful for the patients. There is a clear dose effect with on-chart recovery for 68% and 53% for the first and second affected eyes of bilaterally treated patients. These percentages are higher than the ones for unilaterally treated patients. 69% of bilaterally treated patients achieved at least 15 letters improvement from nadir, and 64% of unilaterally treated patients, which shows a level of improvement of 15 letters or 3 lines, as it is considered to be clinically meaningful. It is important to point out that most of the patients respond to treatment and the results are not driven by few outliers results. Now we are moving to safety. We have a lot of follow-up data with the product. If you include the phase 1-2 study, close to 200 patients were treated with Lumivoq. Lumivoq is well tolerated. There are no systemic safety issues as the biodissemination in the blood is negligible. The main ocular adverse event is intraocular inflammation, which is classical with gene therapy. The good thing with Lumivoq is that the intraocular inflammation is mostly mild, responsive to conventional treatment, mostly corticosteroids eye drops only, and is resolutive without sequela. The good safety profile is comparable in unilaterally and bilaterally treated patients. In conclusion, as the regulatory pathway has been different between EMA and FDA, we filed for EMA registration in September 2020 based on reverse and rescue pivotal studies results. We will then be moving to BLA for US registration in Q2 2022 based on Reflect pivotal study results. I would like to thank patients, investigators, and teams who have participated in these clinical studies. Thank you for your attention.
Okay, yes, but I cannot turn on the camera. It says that okay. Now it's okay. It was blocked. No. Okay, but okay. So we can go on with Matthew Klein, that is a Chief Development Officer, PTC Therapeutics. Please, Matthew.
Thank you very much. As always, it is a pleasure and an honor to be able to participate in the Mitochondrial conference. So many of our partners over many years I see either as panelists or participants. And so again, very grateful to share this update on our development of vatiquinone, also known as PTC 743, previously EPI 743, and our recently launched trial in patients with mitochondrial disease and seizures. For those of you who don't know PTC, we are a global diversified rare disorders company with over 1,200 employees with headquarters in Europe and the United States, as well as several other locations worldwide. Since our founding in 1998, we've been dedicated to using good science to develop innovative therapies for patients affected by rare disorders. And these efforts have resulted in a diversified pipeline with programs at various stages of development. Today, of course, I'm focusing on our BioE or bioenergy platform, and in particular, our drug vatiquinone, which is in development for mitochondrial disease. As I mentioned, vatiquinone, previously known to many of you as EPI 743, is an orally available small molecule that targets the enzyme 15-lipoxygenase in order to decrease inflammation, oxidative stress, and address the glutathione depletion and cell death that characterizes many diseases, including mitochondrial disorders. As I mentioned, is orally available in both a solution form and a capsule form. We've collected a number of orphan designations and other regulatory designations over the years of its development, and have treated over 500 patients, with the longest exposure being over 10 years. And importantly, the data continue to show that the drug is safe and well tolerated. Currently, we are in the phase 2/3 trial for mitochondrial seizures, which I'll be talking about today, as well as a phase 3 trial for Friedreich ataxia, which is also a global trial. Our rationale for developing vatiquinone for refractory mitochondrial seizures or mitochondrial epilepsy has several important components. First, seizures are a highly morbid aspect of mitochondrial disease, and it's been reported that 40 to 50% of all patients with mitochondrial disease have associated epilepsy, which is frequently refractory to traditional anti-epileptic medications. We also have a very strong scientific rationale in that the vatiquinone target pathway has been linked to refractory epilepsy syndromes in both in vivo and in vitro models. And finally, in a number of clinical studies over the years, we have been able to demonstrate that vatiquinone disrupts refractory status epilepticus, decreases overall seizure frequency, and as previously reported here from our work in collaboration with the team at the Bambino Gesù Hospital in patients with PCH6, we have decreased seizure-related morbidity as well as mortality risk. So all of these components together really brought us to the decision to launch this trial of vatiquinone in patients with mitochondrial disease and associated seizures. I'll just briefly mention that we have recorded treatment effects in a number of our studies in a number of different mitochondrial disease subtypes, including decreased seizure frequency and resolution of status epilepticus in patients with Alpers-Polg, as well as demonstrating resolution of seizures and decreased seizure frequency in other patients with MELAS, along with other benefits of treatment, including improved neuromuscular function. We've been also able to look back in our previous trials of vatiquinone and Leigh syndrome, and by looking at this specific item covering seizure frequency in the Newcastle scale, also record an impact on seizure frequency. So again, we have brought all of this experience to bear in our trial for vatiquinone and mitochondrial disease seizures, what we're calling our MIGHTY trial. We've also been able to leverage a number of the lessons we've learned and others have learned over the years in developing drugs for mitochondrial disease patients, as already been discussed by many of my colleagues on this panel today. We know there are a number of critical challenges that have made the development of drugs for patients with mitochondrial disease very, very challenging. As we developed this trial, and quite honestly, as we thought about even doing a trial like this, we wanted to be sure that we incorporated a lot of the lessons that we have learned along the way. First, by using an endpoint such as seizures, which is measurable, somewhat objective, though obviously it has some reliance on report from the parent. We're addressing one important concern, which is that objectively measured endpoints or quantifiable endpoints are often much better than composite scales, both in terms of detecting treatment effect, and also it is well established that regulatory authorities, including EMA and FDA, are much more comfortable with these types of measurements rather than composite scales. We've also addressed the challenge that we all have in designing mitochondrial trials. How do you manage supplements? Virtually all patients with mitochondrial disease are on supplement regimens. They can vary from center. They certainly vary country to country. And in this particular study, we made the decision to allow all patients to remain on a stable supplement regimen that they may have been on before the trial. We have also learned that it's very, very important to minimize barriers to travel sites. And this has been increasingly important in the COVID pandemic. And one way we sought to address this was by including many more study sites, both within countries and across countries worldwide. And finally, there is a challenge we all know, which is disease heterogeneity. There probably is no way to completely address this challenge, though we have focused on a symptom that's common to many different mitochondrial disorders, though they themselves may be heterogeneous. And we've also done things like stratifying randomization to try to ensure that we have balance in both placebo and treatment arms of many different mitochondrial disease subtypes. Again, I don't think anyone can be convinced that you're ever going to fully address heterogeneity, but we have made efforts to try to reduce the impact of heterogeneity on the trial. So let me provide some details on this trial. It's a phase 2/3 trial that's being conducted globally. We have study sites in the EU, including 2 in Italy, as well as sites in the UK, United States, Japan, and Australia. It's a randomized double-blind placebo-controlled trial in which we're targeting to enroll 60 subjects. The overall duration of the first part of the trial is 7 months with a long-term open label extension. The 7 months includes a 1-month observational run-in phase where a baseline frequency of motor seizure occurrence is captured. Then there's a 6-month double-blind placebo-controlled phase, after which all patients will receive treatment for at least 48 weeks. The key inclusion criteria include age under 18 years at time of randomization, the genetic confirmation of mitochondrial disease. There needs to be seizure activity despite treatment at some point in life with at least 2 anti-epileptic therapies. There needs to be a documented history of epilepsy that is associated with the mitochondrial disease. There's also a need to have a stable dose regimen of anti-epileptic therapies prior to the baseline visit. Again, just to make sure that we minimize as many variables as possible once therapy starts. And finally, as I mentioned, they need to have a stable regimen of dietary supplements for 30 days prior to the baseline visit, as well as allowance of a ketogenic diet, which must also be stable for a period of time prior to the initiation of screening. The key study endpoints, the primary endpoint is a change in observable motor seizure frequency, and that's going to be as over that 6-month period compared to baseline, with secondary endpoints looking at disease-related hospital days, occurrence or recurrence of status epilepticus, use of rescue medications, and then of course we'll have a number of safety endpoints. I'll end by mentioning some of the strategies we have taken to mitigate the COVID-19 impact on the trial. Of course, there has been an impact. We've seen it both in the time it has taken to get clinical trial sites up and activated. Also, we are noting that, not surprisingly, there is a concern, and in some cases, a hesitation of patients and families to want to travel or participate in clinical studies during this period. We have tried to address this by increasing the number of study sites and achieving geographic diversity, so minimizing the amount of travel that patients may have to the study site. We've also put the opportunities throughout the study to minimize study visits and rather use remote study visits utilizing telehealth, so we can do as many video visits as possible. This has been very, very helpful in addressing the concern of patients and families for traveling. And this was actually also something that we were able to do in part because the primary endpoint of the trial, the reduction in observable motor seizures, is something that's captured using an electronic seizure diary at home. So this has really allowed us to minimize the visits to the study sites. And I think also we see the entire COVID-19 experience as one to accelerate the movement in general for clinical trials to be more remote. And I think that this is going to be very, very important for patients with mitochondrial disease because the burden of traveling to study sites with the frequency that's needed in clinical trials is very, very difficult. And I think has over the years created, in some cases, a bias in who can participate in clinical trials. So I think now that we are being able to use electronic methods or telehealth methods to allow for remote clinical study visits, my hope is that clinical trials in the future for mitochondrial disease patients can be even more inclusive. So let me end by thanking you all again for this opportunity to share an update with you and for your valued partnership over the years. We do have a website for our clinical trial, which is the www.themightystudy.com if you would like some more information. And again, thank you very much.